Let's talk about some ongoing research in the lab. So as you've heard, for 40 years I've been working on tyrosine phosphorylation, where phosphate is added to the phenolic hydroxyl group, the 4 position, creating a phosphate ester. Phosphate esters are intrinsically very stable linkages. You can release them from proteins with treatment in 6-normal HCl by boiling. So this is a very stable bond. It's estimated that a phosphate ester in water in the absence of metal ions may have a half-life of over 100 years. So a very stable chemical linkage. And serine and threonine are very similar. They are very stable linkages. They're easy to study because of this. How, as I pointed out, the 6 other amino acids can be phosphorylated. The 3 basic amino acids, histidine, arginine, and lysine, cysteine, glutamate, and aspartate. These phosphoamino acids are all chemically very unstable. You can't boil them in acid, for instance, and so they're much harder to study. I became interested in the possibility that histidine phosphorylation of proteins might be an important regulatory process many years ago, based largely on the fact that it was known to be phosphorylated in enzyme intermediates. But the reason I became interested in it was that, like tyrosine, histidine is a ring domain amino acid. And it's not a phenolic ring. It's an imidazole ring, which has 2 nitrogens. And in principle, it can be phosphorylated either at the N1 position, closest to the alpha amino and carboxy groups, or the N3 position. And it turns out that N3 phosphorylated histidine is structurally analogous to phosphotyrosine, and in fact one of the first monoclonal antibodies made against phosphotyrosine recognized 3 phosphohistidine in proteins. It was published in 1982. Anyway, this was one reason for being interested in histidine phosphorylation. But as I pointed out, the phosphorus-nitrogen link, the phosphoramidate bond, is very unstable, and it's heat and acid labile. So you can't separate phosphohistidine from a protein with acid hydrolysis. Instead, you have to use alkaline hydrolysis. So a little bit of history on histidine phosphorylation. It was first discovered as a phosphoenzyme intermediate by Paul Boyer back in 1962 as the intermediate of succinate thiokinase or succinyl-CoA synthase. But it's probably best known, if you know anything about histidine phosphorylation, as being essential for the bacterial 2-component signaling systems, in which there are 2 proteins, one of which is a receptor protein in the plasma membrane that responds to a stimulus, perhaps a nutrient or an amino acid. It autophosphorylates on a histidine, and this phosphate is then transferred onto an aspartate residue in a second protein, the regulator or sensor protein, which is often a transcription factor and is activated by aspartate phosphorylation. So 2 non-canonical phosphorylations in series here. But phosphohistidine is found in eukaryotes as well. There are metabolic enzymes that use it as a phosphoenzyme intermediate, phosphoglycerate mutase, an ATP citrate lyase, for instance. But it's not only found as phosphoenzyme intermediate, it's also found in other proteins as a real post-translational modification. For instance, the N-terminal tail of histone H4, histidine 18, is modified by phosphorylation. And that's found in all eukaryotes, from yeast to vertebrates. So what are the kinases? Well, the only two known histidine kinases are these proteins, NME1 and 2, also known as nucleoside diphosphate kinases, A and B. These are housekeeping enzymes that rephosphorylate nucleoside diphosphates using ATP to regenerate the nucleoside triphosphate. These enzymes use a 1-phosphohistidine intermediate, and they transfer that phosphate onto the diphosphate, but they can also transfer it onto histidine residues in proteins. So they can be protein kinases as well as nucleotide kinases. I should point out that phosphoglycerate mutase, the enzyme intermediate, is a 3-phosphohistidine, and there are structures showing these intermediates. In addition to the kinases, there are 3 reported phosphohistidine phosphatases, and I'll mention LHPP in a minute. So to summarize, in bacteria, 2 component systems use a phosphohistidine phosphoaspartate phosphorylase. In mammals and other eukaryotes, there are histidine kinases, 2 of them, use a phosphoenzyme intermediate to phosphorylate proteins on histidine and possibly other residues. 3 phosphatases that can remove that phosphate. So principle then we have a reversible regulatory modification. We know some proteins that are regulated by histidine phosphorylation, particularly this calcium-activated potassium channel, KCA3.1, from the work of Ed Skolnick at NYU, who showed that phosphorylation of histidine 358 in the C-terminal tail of this channel increases channel opening, and I'll tell you a little bit more about that in just a second. Of interest to Bob Lefkowitz, then, the beta subunit of G proteins is also phosphorylated on histidine by the bound GTP, in fact. So what we've learned over the last 50-something years is that histidine phosphorylation is used quite commonly in eukaryotic cells. It can regulate ion channels, G protein signaling, possibly histone function, transcriptional output in the nucleus. And as you'll see, we found histidine phosphorylated proteins involved in many processes in the cell, for instance, in centrosomes and spindle poles. So let me stress, histidine phosphorylation is very unstable, and here's just some chemical studies showing that not only is the phosphoramidate bond very energetic, significantly more than a phosphate monoester, so it's capable of delivering phosphate or donating phosphate to many compounds, but it is extremely unstable in acid. So here's 1 molar HCl at 50 degrees, 3 phosphohistidine and 1 phosphohistidine have half-lives of around 20 seconds. So extremely unstable.
What about pH7?
pH 7, depending on the sequence around it, it's relatively stable. So yeah, you can work at pH 7 or anything higher than that. So I asked a student about 25 years ago now, 28 years ago now, to try and make antibodies to phosphohistidine, thinking that like the antibodies to phosphotyrosine, these could be very useful in studying histidine phosphorylation. And we tried to use chemically phosphorylated histidine polymers to immunize rabbits, and we never got anything. We assume it was because the phosphohistidine was simply too unstable when injected into a rabbit. So we knew we needed stable analogs of phosphohistidine, and we had to wait until Tom Muir and Mike Webb both reported the synthesis of these phosphohistidine analog amino acids. They are phosphoryl triazolyl alanines, and they have a phosphorus carbon, a phosphonate bond, linked to a 5-membered ring that looks a bit like a histidine. It's not really a terribly good analog, but anyway, they made 1 PTZA and 3-PTZA analogs. And we incorporated these into a degenerate alanine glycine random alloglide backbone to immunize rabbits after coupling to keyhole lipid hemocyanin with the goal of making sequence-independent phosphohistidine antibodies just like sequence-independent phosphotyrosine antibodies. And I wouldn't be telling you this if we hadn't been successful. So here are some of the validation experiments. So we use them in tests for blotting experiments. Now, you're probably asking yourself, well, you can't boil your samples in sample buffer, so how could you do these experiments? And the answer is you don't have to boil your samples in sample buffer to run a gel. You can simply incubate your samples on ice in sample buffer. And almost the vast majority of proteins equilibrate fully with SDS on ice. So we didn't boil the samples, and we altered the conditions of the gels a little bit and ran them in the cold room, blotted them in the cold room, and added antibodies in the cold room, all in an attempt to maintain phosphohistidine linkages. So we tested them against NME1, which I told you has a 1p His intermediate, and phosphoglycerate mutase, which has a 3p His intermediate. And you can see the antibodies generated against 1p TZA recognize NME1, but not phosphoglycerate mutase. Conversely, the 3p His antibodies recognize phosphoglycerate mutase, but not NME1. And here's the control. If you heat the sample in sample buffer at 95 degrees, the signal basically goes away. So we use this control routinely in our experiments. We went on to use the spleens of these rabbits under contract with Epitomics to generate monoclonal antibodies against 1p His and 3p His. And here an example of blotting with one of the 1p His monoclonals against a series of pancreatic cancer cell lines. In each case, there is a boiled control. And you can see that the major bands that are detected are NME1 and NME2 themselves, the phosphoenzyme intermediate, although there are some other bands that go away on heating. And if we blot either E. coli extracts or 293L cell extracts with different 3p His monoclonals, you can see there are a lot of bands, many of which go away upon boiling. So we think it's likely that 3p His proteins are more, the 3p His modification is more frequent in cells than 1p His. It's slightly more stable chemically. So we've generated a significant number of unique monoclonals based on variable chain sequence, and we've used them for immunoblotting. I've shown you immunofluorescence. I'll show you one example, immunohistochemistry, which I'll show you subsequently, and then affinity enrichment of p His proteins to look at the p His proteome. We've also learned about the specificity of these antibodies by generating crystal structures of some of the monoclonals as Fab fragments bound to PTZA peptides. This is the work of Rajasri Kalagiri in the lab in collaboration with Ian Wilson's lab at the Scripps Research Institute. Here you can see 1-PTZA. Here's the 1-phosphate bound to this antibody. Here's the 3-phospho-TZA bound to the Fab fragment of another antibody. And you can see, I show you the actual contacts with the complementarity determining regions, both in the heavy chain and in the light chain. You can see very specific contacts with the 3-phosphate in this case. And if you look at how the 1p His and 3p His are bound, you can see they're in different orientations here. So we've used the antibodies, as I said, for immunofluorescence staining of, in this case, growing HeLa cells. You can see in green nuclear structures of some sort that we showed go away with heat or acid treatment. These two dots intrigued us here. They're sort of in the cytoplasm in early prophase cells, and then you can see that the spindle poles stain positive. So this suggests to us that histidine phosphorylation is likely to be playing some role in progression through mitosis and possibly in transcriptional regulation. We've used immobilized forms of these antibodies to enrich proteins from cells lysed at pH 10 in urea to dissociate proteins so we don't pull down protein complexes. And using a Salk comparison, we've identified around 800 proteins that are enriched more than twofold. And we do biological process analysis. Interestingly, a large number of these proteins are involved in nucleic acid-related processes, translation, transcription, splicing, and so we think there's an interesting role for histidine phosphorylation here. And around 100 are involved in what are said to be cell cycle-related processes, consistent with the immunostaining. And we identified a number of the proteins we expected, including histone H4, PGAM, and ACLY, and NME1 and 2. Now, we've used these antibodies in collaboration with several groups to study histidine phosphorylation in cells, particularly with Ed Skolnick's group to study this potassium channel. He'd reported that this histidine 358 here is phosphorylated, but he didn't know whether it was, well, he deduced it was phosphorylated based on mutagenesis, and he did not know whether this was the 1p His or 3p His. And he'd shown that this phosphorylation by NME2 was responsible then for activating the channel in response to T-cell stimulation with an anti-T cell receptor antibody. And this was important for actually the release of potassium through this channel was important for increasing the intracellular calcium. So using our antibodies, in this case with a stably transfected GFP KCA 3.1 in Jurkat cells and blotting, you could see that the channel is phosphorylated on the 3 position. 3p His signal is positive, whereas there's no 1p His signal of the GFP KCA 3.1 channel. And just by contrast, you can see NME2 is phosphorylated on the 1 position, but not on the 3 position. So we know then that the channel is exclusively phosphorylated at histidine 358 on the 3 position. But we don't know how this selectivity is achieved, and that's something we still need to find out. And then what subsequent work from Ed Skolnick and Steve Hubbard showed is that the phosphate on histidine H358 is likely to regulate the channel by preventing binding of an inhibitory copper ion, which chelates the histidine 358 in a 4-helix bundle formed by the C-termini of this tetrameric channel here. Phosphorylation of that histidine blocks the binding of the copper and allows the channel to open. Now, it's proved very difficult to identify sites of histidine phosphorylation by conventional phosphoproteomic analysis for a couple of reasons. Conventionally, phosphopeptides after tryptic digestion are isolated using TiO2 or IMAC affinity resins, because you need the positive charge on the peptides for them to bind. Those are both done under acidic conditions, and this discharges any phosphohistidine peptides. In addition, to put a tryptic digest into a mass spec instrument, the HPLC buffers are all around pH 2. Again, you lose the phosphohistidine. once a p-his peptide gets into the instrument, the phosphate is lost upon bombardment by neutral loss. And so it's really a triple whammy. You can't enrich the peptides, you can't separate them, and you can't identify the histidine residue. So clearly new methods are needed to identify histidine phosphorylation sites using mass spec. Kevin Adam in the lab has developed a method for enriching the peptides at neutral pH, binding to hydroxyapatite to enrich all phosphopeptides, elution at pH 11, and then using immobilized antibodies, a mixture because they all are slightly sequence dependent, and then elution again at pH 11, and then analyzing by mass spec. We still haven't solved the problem of having to use some acidic buffers and the neutral loss, but when he takes a doubly enriched phosphopeptides from, in this case, I think it was 293 cells, and analyzes using modified software, modified max quant software that can identify post-translational modifications, including phosphohistidine, phospholysine, and phosphoarginine. In what is enriched, which is by no means all phosphopeptides, around 20 to 25% of them are N-phosphate linked, and the rest are O-phosphate linked. And I won't take you through that. We've been looking for motifs. But using these new enrichment protocols, we've identified at least 50 new phosphohistidine sites, using some tricks to identify which histidine is modified in the peptide. And Claire Ayers at the University of Liverpool has developed another method for enriching these acid labile non-canonically phosphorylated peptides. She calls UPACs. This paper's available online at bioRxiv, and she's identified a large number of histidine phosphorylation sites. So taken together, this suggests that there is a large hidden phosphoproteome. Maybe to 20 to 25% of all phosphate linked to protein has previously gone unidentified, and it's linked to histidine, lysine, and arginine, and also probably to glutamate and aspartate. And obviously there's a wide open world here of trying to figure out what all of these modifications do. And that raises the question then, what is histidine phosphorylation actually regulating? Because of its chemical ability, even at pH 7, it could just be used as a timer that it slowly dephosphorylates. But then we know there are phosphohistidine phosphatases, and so probably it's more commonly regulated by enzymatic dephosphorylation. To come back to the theme of phosphobinding domains, are there phosphohistidine binding domains? We've sort of looked but haven't found anything so far. But what we are trying to do is to evolve an SH2 domain with Dev Sidhu in Toronto into a phosphohistidine binding domain, so we can then use this in cells as a probe for phosphohistidine. Another possibility is that it works by causing a large charge change. Histidine itself is +1, also thereabouts at pH 7, and when it's phosphorylated it's -2 or thereabouts. In other words, there's an almost 3 charge change. And then, as I've suggested, maybe it can regulate divalent metal ion binding. Many metal ion binding sites have a histidine or a cysteine that can be phosphorylated. So I think Mike Hall is going to talk about this in his lecture later on in the meeting, but I'll just briefly summarize some of the things that we found together on a role for histidine phosphorylation in hepatocellular carcinoma. Mike, as he will tell you no doubt, generated a new model for hepatocellular carcinoma in mice, in which he conditionally knocks out P10 and TSC1 that regulate mTOR signaling in the liver. Particularly, he knocks out TSC1, which increases mTORC1 activity, and P10, the lipid phosphatase, which increases mTORC2 activity. And these mice develop a fulminant hepatocellular carcinoma within 4 to 5 months. You can see many nodules of tumor tissue here. And Mike set out to try and define mechanisms involved. And instead of doing a transcriptome analysis, as most people would do, he did a proteome analysis on these tissues and looked particularly for kinases and phosphatases that are up or down regulated in the tumor tissue compared to the normal tissue. And he noted that NME1 and NME2 are upregulated highly in these tumors, and LHPP, one of the 3 phosphohistidine phosphatases, is strongly downregulated. And Mike and I happened to be speaking at a workshop on the island of Spetses in 2015. And after I had talked about our new phosphohistidine antibodies, we got to talking, and he said, I think we could do something interesting in looking at these tumors because of these changes in NME and LHPP. Here you can see that the level of LHPP is basically lost in the double knockout tumors, whereas NME1 levels are increased. But what was most exciting was that when blotted with our 3p-His antibodies, the level of 3p-His that was sensitive to heating was greatly increased in tumor tissue compared to normal tissue at about 3-fold. And you can see there was strong upregulation of phospho-NME1 and 2. And Sravanth Hindupur, who did the work in Mike's lab, was able to use one of our 3p antibodies to show that the liver tissue has increased immunofluorescence signals with the SC44-1 3p antibody lost upon heating. So this suggested that maybe loss of LHPP was leading to an increase in 3p-His in combination with increased NME1 or 2. And so he re-expressed or overexpressed LHPP in a cell line derived from one of these tumors using an adenovirus vector. And you can see that this strongly suppressed the level of 3p-His. And more importantly, that expression of LHPP reduced sphere-forming activity of these CB1 cells. And that expression of LHPP in the liver of these mice using an AAV vector injected via the tail vein strongly decreased the number and size of tumors in these mice. We've carried out a preliminary study of what proteins show increased phosphohistidine by mass spec in these tumors, and I won't take you through this because we haven't really done anything with them, but of interest is ACLY, that ATP citrate lyase protein that I mentioned right at the beginning that has a phosphohistidine intermediate. It's critical for fatty acid biosynthesis by generating cytoplasmic acetyl-CoA, the precursor for fatty acid synthesis, and many tumors are totally dependent on de novo fatty acid synthesis, and so this could be involved in increased fatty acid synthesis in the liver. Ultimately, though, this is all in mice. What about in human hepatocellular carcinoma? Mike has a collaboration with surgeons in Basel, and he was able to obtain human tumor tissue, blot this for 3p-His and 1p-His, and you can see a significant heat-sensitive increase in 3p-His in these two tumor samples, again a 2 to 3-fold increase. And so our conclusion is that LHPP is acting as a tumor suppressor in liver cancer and reduces the level of 3p-His proteins, and that 3p-His in consequence is likely to be a driver in these tumors. And what needs to be done, obviously, is to identify which proteins are being phosphorylated that are important for the generation of hepatocellular carcinoma. And we and Mike are currently collaborating to try and identify the key proteins. Assuming this is the case, then might be of interest to try and develop drugs that target histidine phosphorylation. The only histidine kinases I've been telling you are NME1 and NME2. These are redundant but essential enzymes with this housekeeping function, and so a generic catalytic inhibitor is unlikely to be successful because of its toxicity. So either we need inhibitors that only block the protein kinase activity of NME1 and NME2, and we don't know how they select their protein substrates, or we need to target something downstream that's activated by histidine phosphorylation. So that's a new area which I think has a lot of promise. We've made some tools that are useful for studying histidine phosphorylation that are commercially available. And I think there's a lot of opportunity here for anyone who's interested in a new area of signaling. So in the last few minutes, I'll tell you about a second project that's much more translational and is work we've been doing on pancreatic cancer. Pancreatic cancer is the third and about to become the second leading cause of cancer death due to its very high mortality rate, largely because it's diagnosed so late. It's basically asymptomatic until very late in the disease, by which time it's almost always metastatic. It has a very characteristic progression, both morphologically and genetically. An activating KRAS mutation is usually the first event, causes a morphological change in the ductal tissue known as pannins, with additional genetic changes, loss of P16R, loss of P53 and SMAD4. These become more pronounced, and ultimately these cells begin to migrate and invade and can intravasate into the bloodstream, extravasate, and form metastatic nodules, commonly in the liver. So like all solid tumors, pancreatic cancer is not simply made up of tumor cells. It has a large component of normal stromal cells. It has blood vessels, it has cells from the immune system, parasites, etc. In addition, it has a large population of mesenchymal cells, sometimes called cancer-associated fibroblasts, and the pancreatic tumors have a particular type of stromal fibroblasts called stellate cells. And these cells all interact and talk to one another, and the stromal cells support the tumor cells and vice versa. The stellate cells are a resident population in the tumor. They have this characteristic star-shaped morphology, about 4% of all cells in the pancreas. And upon activation, either through tissue injury or tumor-induced inflammation, or pancreatitis, these cells transform into an activated myofibroblast. They proliferate and secrete a large amount of extracellular matrix protein and a variety of cytokines. The pancreatic tumor tissue is sort of characterized by nodules of tumor cells surrounded by this massive stromal structure with a lot of collagen and other matrix proteins and activated stellate cells. So people have argued that it's this desmoplastic reaction and barrier which makes pancreatic cancer difficult to treat. And as part of a Stand Up to Cancer dream team that I joined back in 2010, we became interested in the role of these stellate cells in pancreatic cancer, in particular in paracrine signaling between the stellate cells and the tumor cells and vice versa, with the idea that if there were factors produced by the normal cells, or at least normal genomically stellate cells, we might be able to target them therapeutically. And I'm going to tell you in the next few minutes about our discovery that leukemia inhibitory factor, or LIF as I'll call it, is made by the stellate cells and acts on the tumor cells to maintain them in a stem-like state. So to look at paracrine factors, we use cell lines grown in 2D. I agree that neither of those things is perfect, but that's what we did. We used a human pancreatic tumor cell line, MIA-PACA2, and we used a human pancreatic stellate cell, which in culture is activated, and analyzed the secreted proteins of these cells in serum-free medium by mass spec, and this was particularly a collaboration between Yu Shi, a postdoc in my lab, and Ruijun Tian, who was at the time a postdoc in Tony Pawson's lab. I should say that Yu and Ruijun met in high school in Mongolia, and they have maintained contact ever since. They set this collaboration up quite unbeknownst to me. Anyway, the secretomes of these cells are extremely complex, around 2,500 proteins secreted by the MIA-PACA2 cells and almost 2,000 by the stellate cells. We look for proteins unique to the two cell types, 400 or so from the tumor cells, 170 from the stellate cells. And now you have this long list, and the question is, how do you prioritize what to look at? So just as validation, you can see the stellate cells make a lot of extracellular matrix proteins and actually proteases, etc. And we were very interested, though, in growth factors and cytokines. And I'll just tell you about the stellate cell secreted factors. And we focused on LIF because it was unique to the stellate cells. It's a cytokine that's a member of the IL-6 family. So what we noted was then that the conditioned medium from the pancreatic stellate cells stimulates phosphorylation of STAT3, which lies downstream of the IL-6 family of cytokines at tyrosine 705. We found that when we stimulated tumor cells with conditioned medium, that STAT3 was bound to the LIF receptor. And we found that the stellate cell secretome in testing several stellate cell preparations contains high levels of LIF. And I'll just show you one piece of evidence here. If we use a neutralizing antibody against LIF, we can block most of the phosphostat-3 signal induced by conditioned medium by treating the conditioned medium with the LIF antibody. We also showed that if you knock out the LIF receptor in these tumor cells, KP4 tumor cells, that the signal obtained with conditioned medium is severely decreased. And using an ELISA assay, we could show that the stellate cells have significant levels of LIF. Most tumor cells have make relatively low levels of LIF, including the cell we did the secretome on, MIA-PACA2, but there are some cell lines like CAPN1, CAPN1, that does make a lot of LIF. So the LIF may be produced both by the stellate cells and by the tumor cells. So LIF signaling occurs through the LIF receptor, which is a heterodimer between a LIF binding protein and GP130, which is the signaling subunit common to all of the IL-6 family members, actually, and some additional cytokines here. So each of them has a unique ligand or cytokine binding subunit and a common signaling subunit. And LIF was of particular interest to us because it's a stem cell factor. Many people use LIF to maintain the pluripotency of ES cells in culture. And so this definitely caught our attention. And so this is the signaling pathway downstream of the LIF receptor, which activates, like all cytokine receptors, the JAK tyrosine kinases, leading to STAT phosphorylation, which is a latent transcription factor in the cytoplasm. It migrates into the nucleus as a dimer and drives expression of, among other genes, stem cell genes like SOX2 and OCT4. So the important question was then, is LIF playing a physiological role in pancreatic cancer? And for this purpose, we used the mouse model of pancreatic cancer, the so-called KPC model, in which a Cre driver, in this case PDX1 Cre, which is expressed in pancreatic ductal cells, switches on expression of an activated mutant RAS and deletes p53, which is floxed. And these mice, when both alleles of p53 are floxed, develop pancreatic cancer and extremely rapidly and die by about 50 days on average. So we could use these mice together with a neutralizing LIF monoclonal antibody, which is D25, originally generated by Genentech for a cardiovascular project, but then deposited in ATCC, so it's freely available. We started by showing that if we treat mice, KPC mice, at 25 mg per kg of D25, that we could reduce the STAT3 signal. So the brown stain here is keratin 19 expressed in the tumor cells. And here's a normal IG control and the anti-LIF treated sample, but I know you can't see that, so I've blown that up. And you can see this nest of tumor cells here lacks the brown staining of phospho-STAT3 in the nuclei of these cells. So 25 mg per kg of the LIF antibodies strongly reduces STAT3 staining in the tumor. So we could set up a preclinical therapeutic treatment protocol in which we treated KPC mice starting at day 32 of age with a combination of an anti-LIF antibody or a control IgG and gemcitabine, a nucleoside analog that is current standard of care for treating human pancreatic cancer. So you can see they have slightly different administration schedules, 2 or 3 times a week. And what we found was that the gemcitabine plus anti-LIF gave us a significant increase in survival of about 20 to 30%. Now, this is a very aggressive tumor. It's hard to treat, but this was certainly encouraging. And if we use a slightly different preclinical protocol, which sort of mimics what patients are currently pancreatic cancer patients are currently treated with, which is a triple chemotherapy with nab-paclitaxel. This is an immobilized form of paclitaxel, a microtubule drug. cisplatin and gemcitabine, followed by gemcitabine and an anti-LIF antibody compared to control IgG, you can see we now got a quite significant increase in survival of these mice. So what is the LIF blockade actually doing in these mice? Well, if we look by histopathology, 3 different pathologists at different institutions all came up with the answer that the LIF treatment reduces, or rather increases, the fraction of the tumor tissue which is moderately differentiated. And consistent with that, we found that LIF treatment reduced the number of tumor cells with stem-like markers like CD133 and greatly reduced the number of tumor cells able to grow as spheroids in this panel, or form tumors upon flank injection in this panel here. So it appears then that the blockade is promoting tumor differentiation and possibly reducing the number of stem-like cells. So to corroborate these data, we also generated a genetic model of LIF antagonism by crossing the KPC mice with floxed LIF receptor. And the LIF receptor then is going to be knocked out in the ductal cells. And I wanted to point out that when we used RNA hybridization, RNA-scope, to analyze which cells express LIF on the one hand and which cells express LIF receptor in the tumor tissue in the mouse, the red color here shows that LIF is predominantly in stromal cells next to tumor cells, which are marked by the blue keratin 19 RNA. Whereas, particularly in the tumors, the LIF receptor in red here is present in the tumor cells, not in the surrounding periostin blue positive stromal cells. So the LIF receptor is predominantly in the tumor cells. And consistently then we found that knocking out the LIF receptor in the ductal tumor cells, particularly in mice treated with gemcitabine, gave a significant increase in cell survival. So this argues then that the LIF produced either by the tumor cells or more probably by the stromal cells is acting on the tumor cells in these tumors. Conditional knockout of LIF also reduces the number of stem-like cells in these tumors. So when we carried out a time course to look for when LIF is expressed in the tumor tissue, we could see a steady increase in the level of LIF. We could also detect LIF in the serum of these mice. Just a couple more slides. And again, what was important is this true only in mice, or is it also true in human tissue? And by looking at tumor samples, we could see there were high levels of LIF in pancreatic tumor tissue by ELISA, and we confirmed this with a mass spec assay carried out by Ruojian Tian. And we could also detect LIF here in patient sera. So I'll skip these. LIF levels in the human tumor samples rose in correlation with the differentiation status. So highest LIF levels were found in the poorly differentiated human tumors, and there was a correlation with survival time. So to conclude then, LIF appears to be a reasonable target for pancreatic cancer therapy. It increases survival in a mouse model of pancreatic cancer. There's a correlation between LIF levels and disease state. We can detect LIF levels in serum as well as in tumor samples, suggesting that we might be able to use LIF as a biomarker as well as a cancer therapeutic target. And obviously the next step then will be to develop a humanized anti-LIF monoclonal antibody for testing in human pancreatic cancer. And Northern Biologics, a company in Toronto, with whom I have no financial interest, has developed such an antibody and began cancer trials in August, including in some pancreatic cancer patients. So let me finish then by thanking the people who did the experiments, the work on the phosphohistidine antibodies and studies with those, spearheaded by Steve Foos with help from other members of the lab. Jacques Marget at Sanofi did all the chemistry to produce the antigens with TZA analogs, and all the work on liver cancer was done in my cause lab with Suravat Hindapur. The work on LIF in pancreatic cancer has been a collaboration between Yuxi in my lab and many other people, and particularly Ruja and Tian, and people who provided pancreatic cancer samples, particularly Tim Donahue. We're trying to make phosphorylation antibodies, so keep tuned to that. And we're going to celebrate the 40th anniversary of tyrosine phosphorylation at a meeting in 2020 at the Salk. And finally, this is what you have to do to raise money for pancreatic cancer. Thank you very much.